Alrighty, we've come back to do polygenic inheritance. Okay, with polygenic inheritance, poly, the word means many, genic, you know that means what? Absolutely, genes. Okay, so many genes in order to make the particular trait. All right, so it says Mendel, we know everything he studied, one dominant allele, one recessive allele, one trait, one gene. Polygenic inheritance, it says, is the inheritance of one characteristic, but it involves the action of many genes. Many human characteristics are inherited this way: eye color, height, hair color, skin color. Some examples. So what we want to look at is human skin color as an example. We know the authorities believe that there are 5 genes that are involved in the inheritance of skin color, but we're only going to look at 2 of those genes. The first controlling the amount of pigment that is being produced, and that gene is going to have 2 alleles, and those 2 alleles are the capital A, which is going to say that we are producing much pigment, lowercase a saying little pigment being made. Gene number 2 is controlling what pigment is produced, and that one also has 2 alleles. And so for it, we're going to say capital B, starch pigment being made or produced. The lowercase b is going to suggest that light pigment is being made or produced. Okay? Now, let's go on further. Epistasis occurs between these genes. Okay, so we've got many different genes working together to produce one characteristic or one trait. One of the things that we want to try to remember is that the more dominant alleles, because remember we're looking at skin color, so the more dominant alleles present in a genotype, the darker the skin coloration is going to be. For example, if we have 4 dominant alleles, that's going to be darker than someone that has 3 dominant alleles, which is definitely darker than someone that has 2 dominant alleles. One of the other things that we want to, because we're counting the number of dominant alleles, we want to be aware that if someone has 2 capital A's and 2 lowercase b's, and then the other person has 2 lowercase a's and 2 capital B's, they have the exact same pigment. So if we read it, it would say the first one is making a large amount of light, the second one is making a small amount of dark, but it says those 2 actually produce the exact same color. Why? Because we're counting the number of dominant alleles, so that's important when determining the color range. Now, one of the things that I need to do is work on showing you how to count the number of dominant alleles and determine a particular trait. So we said this is polygenic inheritance. So therefore, we first need to talk about the individuals. Let's just say we crossed two individuals that were pure breeds. We know this is someone that's dark, the darkest being crossed with the lightest. We know that the offspring that are going to be produced are medium, I guess you could say A, B, every last one of them. And just in case you don't remember how to find your gametes, we're going to do that right now. Let's just say if we take two of these, because we know that we're going to have several of those guys, and if we take two of those and perform a cross, so the cross that we want to look at is big A. So two, those that are heterozygous at both gene locations. We want to find our gametes. So remember, gametes are found by using the FOIL method: first, outer, inner, and then last. Okay, so that's going to give us A, B, outer, inner, A, B, and then last is little a, B. And we have the same set of gametes for the second one. So remember, we're going to run these first ones so we can say that this is our sperm, and we'll say these made the, the ovum. So that's a weird mess I made over there. Alright, so I'm drawing the box. And I'm going to put my gametes on the box. My offspring will be inside of the box. So we've got A, B, lowercase a, big B, and little a, little b. Same thing, because remember we said sperm, eggs. Okay, and we know that an egg doesn't, secondary oocyte does not become an egg until it's been fertilized by the sperm, but this is terminology that we've used forever. So, big A, big B. Let's divide this box. Big A, little b, little a, big B, little a, little b. Remember to keep your A's together and your B's together. What happened there? Okay. Grab my pen back. Okay, and then this one is big A, little a, big B, little b. Big A, big B, little b, big A, little b, little b. Remember, just dragging across and dropping down. Big A, little b, big B, little b. Big A, little b, little b. Big A, little a, big B, big B. Okay, now that we've got the box filled in, we want to do a genotypic ratio. And then we're going to use the genotypic ratio and figure out our phenotypic ratio. So genotypic ratio. All right, so starting with the first box. I have discovered a problem. There we go. Sorry, gang. 2 big B's. I knew something was wrong, though, when I saw it. So big B, one of those. And then big A, big B, little b. Then big A, and I failed myself. Then we got a repeat here. So that's big A, big B, little b. And then big A, little b, little b. We got a repeat here. Got another repeat that's here. And then little a. Big B, one of those. Then little a, big B, little b. Then we got a repeat here. We got a big A, got a repeat there. We got a repeat here. And then we got this by itself. So I end up with the ratio of A1 to 1 to 2 to 4 to 1 to 1 to 2 to 1 to 2 to 1. It's my genotypic ratio. Now let's work on the phenotypic ratio. Now remember, this is polygenic inheritance, which says many genes act on one trait or characteristic. So phenotypic ratio says I've got to base this on the number of dominant alleles. So let's start with the 4 dominant alleles. 1, 2, 3, 4 dominant. There's one of those. Then we need to go to the ones that have 3. So that's big A, big B, little b. So far, I see one of those. Aha. I made a mistake. That should have been a 2 because this is the same thing. So that's going to change the second one to 2. And following the 4, there's no 1. So I'm going to take that out. Now that looks better. Okay. Did I make another mistake like that? This is not normal. It's too early in the morning. All right. So there are 2 of those. Are there any more with 3? Yes. Big A, little a. Big B, Big B, it says it's 2 of those. Okay? So we've done that, that, and that. Then it says, well, how many have 2 dominant alleles? And that would be big A, little a, big B. And there's 4 of those we can see. 2 dominant alleles, so big A, little b, and little a, big B, big B. And then we gotta go to the next category, which is 1 dominant allele. So down to here. So big A, little a, big B. There's 2. Little a, big B, little b. It's 2 of those, and finally little a, little b, little b. One of those. So the ratio, look, is a 1, that totals 4, that totals 6, 4, and 1. So the ratio is a 1 to 4 to 6 to 4 to 1. And we want to say that this is darkest, dark, okay. So you'll see that you can see that even though this looks like a dihybrid cross, we want to treat it differently. So let's get the rest of these. So dark to medium to lightest. Okay, that's the end. And let's go back to the, well, if we went back to the previous slide, it says width, height, and color and eye color. Usually what you're going to get is this bell-shaped curve. And if we've charted that 1 to 4 to 6 to 4 to 1, that's exactly what we get. The 1, say the 4, the 6, and then 1. So yes, a 1 to 4, 6, 4 to 1. And that gives us a bell-shaped curve. Alrighty, hope you enjoyed that little short example of polygenic inheritance.